This is for the qualitative carbohydrate analysis lab. So there's some information here that you're obviously going to read and go through. Um, Let me get to the first page here, a little ahead. So we're looking at this formula, carbon, two hydrogens, oxygen. So for every carbon, there's two hydrogen. For your oxygen, there's also two hydrogen. So it's asking you the question of what actually falls into this formula here, where there's twice as many hydrogens as there is compared to the carbon and comparing to the oxygen. So if we look here, 6C6H12O6, that meets the formula. C12, well, that's not twice as many. But 22 hydrogens, half of that would be C11, right? So this particular one doesn't really fall into place. C5H10O5, yeah, meets that formula when we're looking for a carbohydrate. So are all the following formula correct carbohydrates? Now it seems that there's not. There is one outlier there. And then the rest. So we're looking at, ketones, aldehydes, aldoses, ketoses, and you'll do some answer some questions here. And you'll be looking at the functional group. I did want to kind of explain this, what was going on here. So this question here is asking about the bonding positions for this particular carbon here, right? The alpha, I'm sorry, the anomeric carbon right here, or anomer. So here, to one position, there's an oxygen. To another position, or the second position here, we have the hydroxy. We have another third position here, and there's another carbon. And this is the fourth position. So the fourth bonding position on this shared carbon is occupied by this hydrogen. So that's what they're asking for in this question here. Here, this particular, you're going to look at it the same way, right? Here's one bonding position, another bonding position where the oxygen, one has another carbon, and then this one is also that fourth bonding position is shared by another carbon. So that'll help answer that question there. So a lot of this analysis that you're going to be doing today, keep this page open here. So you want to click on here. It's on page 11. Click here, and you're going to want to keep this open because you're going to answer questions regarding this information here after you've done the experimental portion to it. So there is a lot to the experiment. Okay. Here's a chart that you're going to need to draw out. Okay. So you're going to draw this one out, and you're going to insert positive, what a positive and negative result looks for each one of these reagents here. Molish, iodine, Benedict's, Barfed, Solomonoff's, and Biles tests, and what a positive and negative result look like in your own notebook, so that we can narrow down what this particular unknown is. So as you go through this, right, I have it for the first one here. Now, what I like to do here is you're going to grab these pipettes here. Don't use the same pipette for different solutions. One pipette, one solution. I'm just going to hover it here, and you see it sitting on there. And then the other one here is sitting on Molish. Now, you can run out of reagents, so try not to waste it. If you run out of reagent, sorry, you have to restart it. Right? We can't be wasting these reagents because you're going to see these levels go down as I pull it out. Okay, so um, and you don't need to fill up the test tube up to the top. Just a couple drops of this, couple drops of this, it will be fine. And drag it over, and you'll see that there's the slightest amount in there again, right? And now I've pulled it away, put it back, and it drops another one there. Now I'm going to add the glucose into there. You'll see it goes up even higher. And again, I pull it away, and I'm going to drop it back in there, and it adds it even higher. So just a couple drops of each one. You could even do one and one. It's okay. Um, I'm going to put it on here and I'm going to mix it, and we see a color change here. And it tells you and it describes in here. So a positive result is a pink and violet color. And if that was negative, it would look like this. But also make sure that you're writing this down. Um, so for this test here, um, a positive test indicates that the sample does contain a carbohydrate. Doesn't say whether it's an aldose or a ketose. Mm. Just that it is some carbohydrate. Okay. And you're going to be doing all of these. So you're going to be testing all of these reagents here, right? Um, I'm sorry, all of these reagents. So the moles here, the iodine, benedict. So you're going to be testing all of these reagents with glucose, starch, ribulose. So there's going to be quite a bit of information there. But follow the directions there. And then at the very end of this, there's no calculations that you need to make. This is strictly qualitative. You're just going to answer the questions here. Once you do have your chart ready, right, you're just again going to be answering a bunch of questions regarding the unknown and the starches here. So you should be able to get through that relatively easy. Again, it's strictly qualitative. There are no calculations that need to be made on this particular lab.